Thank you very much, Chris. Dr. Socius, it's a real pleasure to be here with you this afternoon. And I want to thank the organizers for giving me the opportunity to talk to you about this important topic of progress and challenges in HIV prevention. And the approach that I'm going to take is that from both a non-vaccine and a vaccine standpoint. But before I get started, I want to just take a 40,000-foot look at what I believe everyone in this room has as our common end game, and that is the ending of the HIV/AIDS pandemic within our generational time. Now, I'm going to start off with this, and I'm going to end up with this. But the way that we can end the AIDS epidemic has everything to do with driving down global incidence. We won't do that in a uniform fashion because of the disparity of incidence throughout regions of the world. But the end game will be to drive down incidence. And as shown on this incident map, which I will come back to a couple of times during the talk, clearly there is a lot going on. We know that despite a lot of favorable results, we still have a number of challenges here. And they're disparate, as shown by the relative darkness and lightness distributed throughout various regions of the world. And what I'm going to talk about this afternoon is the pathway to the decrease in incidence will be a comprehensive global HIV prevention agenda. And I'm going to divide it into three areas: a non-vaccine, a vaccine approach, and then close up with discussing something about the implementation gap that challenges us. So let's start off first with non-vaccine prevention. The fundamental basis of non-vaccine prevention is HIV testing. Now, when we talk about testing, almost reflexly, we think about testing, finding out if someone is positive, and if they are, put them in the care continuum that we have discussed in great detail at this meeting and at many prior meetings. But given the fact that I'm talking about prevention, I want to focus about the prevention continuum when someone tests as negative. Because as a matter of fact, testing negative once is not the end of the game in HIV prevention. And I adapted this slide from a paper from a couple of years ago by McNary and Elsada, in which they make a very important point. There's a strong analogy between the care continuum and the prevention continuum. As I said, if you test negative, it's not all over. You have to have continually counseling, and according to your risk stratification, you are provided with a rather robust prevention toolbox, which I'll get back to in a moment, that is tailored to the particular risk that you're in. And very similar to the care continuum, you want to retain people in preventive services because it's not a one-shot deal. That's ongoing counseling, support, and outreach. And just like with treatment, there's adherence, and we need adherence support, repeat testing where appropriate depending on the risk. Now let's focus in on that middle box, the prevention toolbox. As you see on the right-hand side of the slide, we have a number of prevention modalities, some behavioral related, some medically, and some both. Now, rather than go through each and every one of them with you, and I'll get back to a few of them as we get on to the talk, what I want to discuss in a little bit depth is the role of prevention in several of these, the role of treatment in several of these prevention modalities. So I've highlighted them in yellow, and I'll talk a little bit about them, from mother-to-child transmission, to PEP, to PrEP, to microbicides for women, but also to treatment as prevention, which has already been a major topic of this meeting so far. So if we're talking about treatment, we have to go back a couple of decades to the fundamental basic research that was done on the viral biology and delineating the replication cycle, which over the years led to really the birth of targeted antiretroviral therapy, to the point where we now have, as we all know, and we've all used, those of us who take care of patients, these drugs in various combinations. And I can't help but reflect 19 years ago, literally in this location, and I know many of you, but certainly not all of you, were in the room at the time when we had what we didn't fully realize until a little bit later was the transformation and the complete changing of our approach to the HIV/AIDS pandemic. There were a number of papers. I remember very clearly sitting in the audience when Trip Gulick, in a late breaker on July 11th of 1996, presented the paper using the first protease inhibitor that was in that trial together with AZT and 3TC. Subsequent to that meeting, there were a number of papers, 3 of which are here as representative papers, that all of a sudden turned around everything that we did. Now, that was a transforming period in the history of HIV, and I'm going to get back to that now as we are here returning in Vancouver. So very shortly after those papers, there was a succession, rather rapidly, of a number of new drugs, which led to what we have now is a comprehensive combination therapy, sometimes with one pill, that has now been able to change things in a global way. So as long as we're talking about therapy, the obvious applications of therapy are in treatment. I'm not going to talk about treatment for treatment's sake, but I want to talk a little bit about the role of therapy, antiretroviral therapy, in prevention. And I'm going to take it from 4 standpoints. The first, prevention of mother-to-child transmission, and there's a strong and really very deeply scientifically based history to that. Starting off with the classical ACTG 076 study, the first time showing that antiretroviral therapy, even with a single drug, to a mother for several weeks during pregnancy, followed by the same for the infant following birth, led to a dramatic diminution in mother-to-child transmission. But we've come a long way then, with various iterations over the years, for shorter courses to more robust courses, to the point now, if you trace that history from the 1994 076 to the point where we now have B+ for mothers, to where we are right now in 2015, as I'll get to in more detail when we talk about the various studies that have just recently come to light, we really need lifelong therapy for all, including pregnant women, which will clearly end the mother-to-child transmission issue that we all face throughout the world. The results have been striking. In the 1990s, there were about 600,000 babies born who were infected from mothers who were infected. There's been now a 60% reduction to the latest count to about 220,000 since the first ACTG 076 study. I'm going to get back to that 220,000 in a few minutes. What about pre-exposure prophylaxis? Again, a robust scientific history. If you look at the science and look at the data, there is no doubt that pre-exposure prophylaxis is highly, highly efficacious, particularly but not exclusively in men who have sex with men when taken as prescribed. A number of studies have nailed that down. We still have a challenge with the effectiveness with regard to women, probably from both a biological standpoint and to some extent from an adherence standpoint. But when you're dealing with pre-exposure prophylaxis, there's always the issue of adherence, and can we as scientists in the scientific community do something about that? And I don't mean just from a behavioral standpoint. And what we're doing, as you're all aware now, is putting a big push for the development of long-acting injectable antiretroviral drugs to prevent HIV infection and maybe even to be part of the treatment of HIV-infected individuals. And there are a number of trials that will be either ongoing or will be ongoing soon. With regard to women, the intravaginal microbicidal rings is something that is now being very actively pursued. It gives women control over their sexual health. It can provide lasting, but importantly, reversible contraceptive, and there are multiple human trials underway. Post-exposure prophylaxis. Interestingly, that this is likely a rather underutilized modality of prevention. And when one tries to figure out why that's the case, you look at some of the challenges that I think are highlighted on these two papers. It's a challenge of awareness that it's actually available, and adherence once you start it. And as you see on the paper on the left, only 36% of high-risk individuals in a study in New York City were actually aware of PEP or even PrEP. And then if you look at the study, the meta-analysis on the right-hand side, 43% of eligible individuals actually failed to complete a PEP regimen. So we do have some significant challenges if we are going to utilize that as one of the things in our prevention toolbox. And then there's the now highly discussed treatment as prevention. And this has been just a very important positive aspect of where we've been over the last few years, dating back to now that classic study of HPTN-052, which you're all familiar with, showing in serodiscordant couples that if you start between 350 and 550 of your CD4 count, as opposed to waiting to 250, there's a 96% reduction in transmission. And as you've heard from my Cohen and colleagues, if you come back a couple of years later to today, those results still remain very robust. The question that was arisen with those original studies is that that may have a good public health impact, but is it good for the people who are taking the drugs earlier? And we'll get back to that in a second. But before we do, let's take a look throughout the world in just a few representative places. If in fact you implement treatment as prevention, you do decrease the incidence. So as you see starred on this map, my incidence map, 4 places which we'll take a look at some representative data. These are just a few of the studies that are available. First, let's take a look at San Francisco. It's very clear that when viral suppression rose from 45 to 78%, the incidence fell by almost 50%. In Rwanda, for every 10% increase in antiretroviral coverage, this was associated with a 6% decrease in HIV incidence. What about Baltimore, Maryland at Hopkins? When viral suppression was increased from 22 to 59%, the incidence in Baltimore decreased from 2.3% to 0.25%. And finally, right here in British Columbia, where the concept of treatment as prevention has been very, very strongly stressed by Julio and his colleagues, when ART coverage rose from 11 to 57%, HIV incidence fell by 42%. So there's no doubt about it that when you look at the effect on a public health-related area, that the data are very robust. But the question arises, as we all know, what about the people who are actually being treated to help prevent infection in others? And we have 2 studies now, one the Temprano study, which took place in Cote d'Ivoire, which clearly showed that early ART, as compared to delayed treatment, reduced the risk of death and serious illness, such as with TB and bacterial infections, by 44%. But now the real nail in the coffin of that is the strategic timing of antiretroviral treatment study, or the START study, which is being actively discussed at this meeting. And as we all know, in about 4,600 adults in countries throughout the world, it was very clear that if you started therapy above 500, which means at any CD4 count for anybody who's infected, compared to waiting until the CD4 count drops to 350 or less, there was a 53% decrease in serious illness or death related to AIDS or non-AIDS. And so if you put those 2 together, the HPTN-052, which has clearly shown an unquestionable public health benefit, together with the START and Temprano studies, which showed an absolute individual benefit, there is no doubt where we are right now. The science that asks for and essentially demands beginning treatment at any CD4 count is as solid as we can get. So there really is no excuse not to act on that scientific information. Let's move on to vaccine prevention. Now, a couple of weeks ago, Science had an issue, the cover of which was devoted to discussion of the progress that we've made towards an HIV vaccine. I'm going to get to that in a second, but I want to just tell you how I look at the vaccine field. I broke it up into a couple of tracks. If we just start off first with the B-cell approaches, I'll get to the T-cell approach in a moment. But if you look at the B-cell approaches, there's parallel tracks. The first is the RV144 approach, which I refer to as an empirical or an inductive approach. And what do I mean by that? I mean, you do the experiment, you get the results, and then you examine the results, try to make a correlate, and then improve on that. It was really quite empirical because, as some of you with corporate memory know, that the RV144 trial, there was some concern and argument about whether it should be done because the individual components of the prime and the boost did not actually induce anything that looked like it would be protective. But on an empiric basis, the trial went ahead, and indeed, to our gratification, it was the first signal of efficacy, about 31% in that trial in Thailand. Now, 31% is not ready for prime time, and it certainly is not ready for wide deployment. However, what it did, as empiric studies do, it laid the framework for an extensive delving into correlates of risk, correlates of immunity. And some very interesting things came out of that trial. We know that IgG antibodies against predominantly the V2 region of the HIV envelope was associated with reduced infection. Interestingly, they were not classic broadly neutralizing antibodies. But these antibodies did have ADCC activity, and it was also shown that there was an interesting reverse correlation of IgA antibodies that actually did correlate with an increase in infection. So now you follow through on that empiric approach. And where do you go from here? Well, obviously, you want to increase the strength, the breadth, and the durability of what we found in RV144. And that's done by a number of approaches: multiple boosts, modified vectors, and adjuvants. And in addition, as we've heard from one of the sessions yesterday and today, there are a number of tracks that are being approached. One we refer to as the licensor track. It's part of a POX protein public-private partnership, which is the reason it's referred to as P5, involving a number of agencies and a number of pharmaceutical companies. The goal of the licensor trial is to replicate the results in Thailand for the support of local licensure in South Africa. Now, simultaneously, approximately with that, will be a more complicated research track, which will assess the impact of different priming, alternate dosing, vectors, and adjuvants to try and get the best possible approach using that broad framework that we did with the RV144 trial. The next one is what I refer to as the broadly neutralizing antibody approach, or the theoretical or deductive approach. So what do I mean by that? Opposite from the empiric approach is to find and to assume where you want to go. What do you want to induce? And then to design your vaccine to induce that. And the reason why broadly neutralizing antibodies come up, because we know, interestingly, from experience with HIV-infected individuals, that these antibodies are detectable in plasma and are elicited, curiously, in only a minority, about 20% of infected individuals. The interesting thing is, which is a real challenge when you're talking about vaccine, which I'll get to in a moment, that these antibodies only appear after 2 to 3 years of continual bombardment of the immune system by actively replicating virus. That's not great news to try and develop that with a vaccine. Most of these antibodies demonstrate a high degree of somatic mutation, which is merely a reflection of the fact that they've been stimulated for quite a while. and certain of them have unusual, somewhat disturbing traits, such as autoreactivity. So this is a crude schematic diagram in the light gray blue in the background of the HIV envelope trimer. And the squiggly figures you see there are some representative broadly neutralizing antibodies that have been extensively studied by a number of investigators. By no means are all of the important antibodies shown on the slide. But the reason I show that is that when you're looking at a vaccine, what those antibodies do is that they recognize what we call neutralizing epitopes on the HIV envelope primer shown here, such as the CD4 binding site, the membrane proximal external region, and on and on. We're going to get back to that in a second, but let's go back to the antibodies. On the road to developing a vaccine, a very interesting conflation has occurred in the field between vaccinologists and people who are looking at treatment as well as prevention in a non-vaccine way, which is kind of ironic, but I think rather beautiful to see the fields coming in from different directions to each other. And let me explain what I mean. Because putting the vaccine aside for a second and look at the potential applications of broadly neutralizing antibodies. First, with regard to treatment. We know from a number of animal studies, and again, I can't bombard you with every study that was ever done, so I'm just pulling out some representative ones. It is shown that passive transfer of monoclonal antibodies in a monkey model clearly suppresses viremia. A recent study from Michel Nutzenzweig lab used his 3BNC117 broadly neutralizing antibody actually in a human phase 1 study in which viremic individuals were given this antibody, which resulted in decreased levels of HIV, which persisted for as long as 28 days. So clearly, this is an antibody that has activity against the virus. What about non-vaccine prevention when you're talking about broadly neutralizing antibodies? You can look at it from two standpoints: passive transfer of antibody as it exists, or gene transfer. So let's take a quick look at where we are now with passive transfer. Again, here's where the animal model helps us. There have been a number of studies from different laboratories in which passive transfer of neutralizing antibodies clearly resulted in the prevention of SHIV infection by a variety of challenge models. In addition, there have been things like the bispecific antibodies directed to CD4 and HIV envelope that David Ho and his colleagues have been pursuing. So the field is really, if I might use that word, very hot. Things are going very quickly from the standpoint of doing it in an animal model to a number of phase 1 studies that are either already going or are being prepared to be launched. The same holds true for gene transfer or vector-based expression of the genes that code for the broadly neutralizing antibodies. And here is a list of just some of the studies from Phil Johnson, from Dave Baltimore, and a recent study from the VRC in which gene vector-mediated antibody expression provided protection in animal models, everything from a monkey model to a mouse model and on and on. These almost certainly will be going into humans in a hopefully a coordinated way to answer some important questions. And then a recent interesting paper from Farzan using a fusion protein against two components of binding sites, both the CD4 and the CCR5. So again, the field is moving in a very exciting, very rapid way. So, having said that now, I couldn't help be struck, having been on both sides of the fence of treatment and prevention and being involved in it, if you look at the framework of the replication cycle with the targets and the HIV envelope, there are really some interesting analogies. Namely, you're targeting a drug to a component of the replication cycle, and you're targeting your antibodies to an important functional component of the HIV envelope. So let me develop that with you a little bit more. Let's take a look at why they're very similar. We have agents that target structure with impact on function. So let's take a protease inhibitor, blocks the protease enzyme, has a major effect on the function of the virus. Let's take an antibody to the CD4 binding site, the same thing. It interferes with the function of binding and helps to block. What about combination? We've been using for years combinations of agents to increase the efficacy and avoid resistance. Integrase inhibitors with non-nukes, protease inhibitors with nukes. The same thing is going to be done with antibodies, and we heard questions about that in one of the sessions today, that there will be combinations of antibodies to try and avoid the resistance, but as with drugs, you're going to have to test them one at a time first. It's going to be the same thing. I think historically, they're going to track each other. What about multiple agents targeting the same site? Two non-nukes against the reverse transcriptase. We're going to have antibodies against the same neutralizing epitope on the envelope. And then lastly, but importantly, just as I mentioned a minute ago, the development of long-acting injectable agents for antiretrovirals, we clearly are going to be manipulating the antibodies so that when you give it, you don't have to give it every month or two. You may be able to give it every 6 months. So there's going to be a lot of activity that's going to be going on, and the analogy, I think, is very strong. So now let's get to vaccine prevention, an extraordinary challenge for all of us. The critical challenge and the fundamental challenge is to take those neutralizing epitopes that I've introduced to you just a few slides ago on the left-hand slide of this slide and convert them into immunogens that would actually induce the broadly neutralizing antibody that natural infection has such a terrible time doing in an easy way. In order to understand that, there are two critical factors that have dominated the field of the induction and the study of neutralizing antibody. One is the stability of the HIV envelope trimer. So what do I mean by that? I'm going to show you a couple of bullets, and then I'm going to give you some schematic look at it to explain better what I mean. The HIV envelope is naturally unstable, particularly if it binds to the binding site. It does all kinds of conformational changes. Upon binding to CD4, for example, one of the things that happens is that you expose an immunodominant V3 part of that envelope. I'll get back to that in a second. The problem is that antibodies to V3 have limited neutralization activity. On the other hand, the stable envelope trimer preferentially binds neutralizing antibodies. So that strongly suggests that the stable trimeric envelope should serve as the preferred immunogen to induce broadly neutralizing antibodies. Let me schematically go through that with you. So if this is the HIV envelope trimer, to understand better visually conformational changes, let me superimpose upon that the molecular conformation of that envelope, and I'll show you an example of what I mean by one of the things that happens with the instability. So if we pull out the crude scheme in the back and are left with this envelope trimer in its molecular configuration, what happens, for example, when it binds CD4 as the V3 loop kind of flips out? Now, what has that got to do with anything? Well, it has a lot to do with it, because as I mentioned just a couple of slides ago, the V3 loop is immunodominant. So it's going to crowd the immune system to make antibodies to it. You would think that's good news. Not really. The bad news is that those antibodies have limited neutralization activity. If you look at the stable trimer, the interesting thing is that, as I mentioned, the stabilized trimer preferentially binds neutralizing antibody. So that would be a preferred immunogen for vaccination. And a number of extraordinarily exciting studies over the last several weeks to a month have, I believe, put us on the track to where we want to go. These are just a couple of examples. A study by John Moore that came out just a couple of weeks ago in Science showed that a stabilized form of the native trimer stimulated autologous broadly neutralizing antibodies in rabbits and non-human primates. Now, you want to do better than autologous, but that's the first step to the broadly neutralizing antibody. So it's not the end game, but it's an important first step. Peter Kwong, John Boscola, and others in a study that just came out again a couple of weeks ago in Nature Structure and Molecular Biology characterized in the fixed pre-fusion conformation the stable trimer, strongly suggesting that this could be used as a vaccine immunogen. Okay, so let's assume now that we got the immunogen in the right conformation. Now the challenge is, how do you engage the proper sequential engagement of the B-cell lineage, which is a significant issue? So let me run through that with you. An experiment of nature, and by saying an experiment of nature, is someone who happened to have gotten infected in a prevention trial, so investigators, Bart Haynes and his colleague, were able to follow that person literally from the time of infection over a couple of years. And what they noted is that you had the development and evolution of broadly neutralizing antibodies that paralleled the mutation of the virus as it was trying to escape the immune response. So what was happening is that after 2 years, the person had broadly neutralizing antibody. Unfortunately, it was too late for that person. But what it did is it showed that as the virus mutated, it was able to induce what we want to induce as vaccinologists. Michel Nutzenzweig, in a review in Science soon thereafter, made the proposal that everyone was thinking about, but he very nicely articulated, is that perhaps we can design the immunogens to mimic a mutating virus and sequentially coax the immune system on to get to the broadly neutralizing antibodies. But hopefully we can do it in less than 2 years, which means you have to probe the B-cell lineage to figure that out. That's exactly what's going on. And that's what a couple of papers have shown is something that's feasible. In a study by Michel Nutzenzweig, he took a series of trimeric envelope immunogens and was able to move from naive germline to broadly neutralizing antibody production in an experimental model. In a study by SHIV and Burton, they tailored immunogens to stimulate germline B cells, creating antibodies with characteristics of broadly neutralizing antibodies. So these are first baby steps, but they're very exciting because they're going in the direction where we feel they need to go. Now, obviously, when you talk about vaccine, you have to consider the possibility, if not the likelihood, that we'll need T-cell involvement. And we all know the importance of T-cell and B-cell function. It's help, it produces antiviral cytokines, it boosts neutralizing potential and potency via T-cell follicular helper cells, but importantly, it can induce cytolytic CD8 positive T-cells. And in that regard, there's a study now, as we all know, it's a very well-recognized study by Lou Picker and his colleagues, in which they used a live vector, a rhesus CMV vector, and in an animal challenge model, all animals became infected. However, 50% of them cleared virus. So the potential of having a live vector continually stimulating might in fact be important not only in vaccinology, but perhaps even in the issue of a cure where you mobilize CD8 positive T cells. And it's just another example of the different phases of what we do of cure research overlapping with vaccine research, prevention overlapping with treatment. It's really a very interesting and lovely phenomenon. So let me close by talking a little bit about the need to close the implementation gap. We really have a serious implementation gap despite all the triumphs that we talk about. Just a couple of examples. 54% of HIV-infected people know their status, which means that 46% don't. So when you're talking about treatment as prevention, we really have a problem there. 220,000 infants were infected in 2014 globally. We have to do better than that. Of the people on ART, the rates of suppression are suboptimal. Even in my own country, the United States, it's suboptimal. Fewer than 1/2 of the targeted 20 million people who should have been circumcised have not actually been circumcised. So we have a long way to go to implement a very, very effective modality of prevention. And also, as we've heard from several speakers, stigma and discrimination persist. One of the things about implementation gap, not the only thing, is this issue of focusing treatment and prevention on hotspots. And when I say focus, I don't mean to the exclusion of other things. But if you look at limited resources, to take a look at how you can get the best bang for the buck. There have been a number of discussions about that. A paper in Lancet from last year by Peter Piot and Mark Dybel brought up the point that if you focus on high transmission geographies and populations and provide tailored packages of intervention, you can actually be much more efficient in both your treatment and in your prevention. So let me give you an example of hotspots. So this is a map of the United States and the typical feature, and this is delineating HIV diagnosis by county in the United States. And obviously dark is bad, light is good. And as you can see, there's a significant disparity. If you pick out one of those states, New York State, within New York State, there are significant disparities. And in fact, using this, Governor Cuomo of New York State has recently instituted a 2015 blueprint with the goal of doing what we want to do globally, is to end AIDS and HIV in New York State by, in a very aggressive way, diagnosing and linking to care, treating with ART. Now that we have the START study, treat everybody with ART as soon as you find out that they're infected, and facilitate PrEP and PEP with the goal, if you can reduce infections from the current 3,300, which was measured in 2012, to about 700 and 750, the mathematical models say that you're going to head towards a diminution, if not stopping, of that pandemic. It isn't only in New York or in the United States. Take a look at a developing country, Kenya. On the left-hand side, Kenya is not the darkest region in southern or eastern Africa. Yet when you blow up Kenya, within Kenya, there certainly are hotspots located around the border of Lake Victoria, which is densely populated. They have low circumcision rates, and commercial sex work flourishes, particularly associated with the fishing trade. Focal implementation of tailored packages of aggressive medical male circumcision and treatment as prevention could actually prevent 600,000 infections by 2030. So that's a little bit of a hint about closing the implementation gap. So let's get back to my incidence slide. So this is the target. I've spoken to you about what the tools are. So if we want to comprehensively prevent HIV, a vaccine is going to be the nail in the coffin of HIV/AIDS. But even without a vaccine, we can go a long way to getting to what we want to do. Because if we implement this comprehensive global HIV prevention agenda, without a doubt, it will lead to a dramatic decrease in HIV transmission. I've already showed you data, and others in this conference have showed you clear data that that absolutely happens. It happens here in British Columbia, it happens in Africa, it happens in the United States. If you do that, there will be a marked decrease in incidence, and that map will lose some of its dark places. And then if you do that over a period of time, we will be able to ultimately end the HIV/AIDS pandemic. This is a goal that is clearly achievable. It's not something of whether we can do it, it's whether we will do it. And the issue is really strong. We are in a very interesting historical point now, just the way we were in 1996, right here in Vancouver. We had something historical. We knew that these drugs worked, and we did it. Right now, we have a challenge of ending the AIDS pandemic within our generation. And our generation has an important historical opportunity to do something that will really shape medical history. Let's not lose that opportunity, or what I call it, a privilege. Thank you.